Hello everyone, welcome back. Today we are going to understand mass spectrometry in a very simple way. Its principle, working and how it identifies molecules. Let's get started. Mass spectrometry, commonly known as MS, is a powerful analytical technique used to identify, characterize and quantify chemical compounds. The technique works by converting molecules into ions, separating these ions according to their mass to charge ratio, and detecting them to produce a mass spectrum. Mass spectrometry is widely used in pharmaceutical analysis, analytical chemistry, bioanalysis, forensic science, and research. In this video, we will understand the complete working principle of mass spectrometry, including ionization, mass analyzers, detectors, mass spectra, and major applications. Basic principle. The basic principle of mass spectrometry consists of four major steps. First, the sample molecules are converted into ions. Second, these ions are separated according to their mass to charge ratio, represented as m/z. Third, the ions are detected. Finally, the detected signals are processed to generate a mass spectrum. Therefore, mass spectrometry essentially converts molecular information into measurable mass to charge information. What is m/z? One of the most important terms in mass spectrometry is m/z. m/z means mass to charge ratio. It represents the mass of an ion relative to its charge. For a single charged ion, the charge is +1, so the m/z value is approximately equal to its molecular mass. If the same molecule carry two positive charges, its observed m/z value becomes approximately half of its mass. Understanding m/z is essential for interpreting mass spectra. Major components. A typical mass spectrometer contains several important components. The first is the sample introduction system. Next is the ion source, where molecules are converted into ions. Ion optics then guide and focus these ions. The mass analyzer separates the ion according to their mass to charge ratio. The detector detects the ion. Finally, the data system processes the signal and displays the mass spectrum. Sample introduction. Before entering the mass spectrometer, the sample must be introduced into the instrument. Depending on the analytical technique, samples can be introduced directly or through a separation technique, such as gas chromatography or liquid chromatography. When gas chromatography is coupled with mass spectrometry, the technique is called GCMS. When liquid chromatography is coupled with mass spectrometry, it is called LCMS. Ionization. The next and one of the most important steps is ionization. Mass spectrometers detects ion, not neutral molecules. Therefore, the analyte molecules must be converted into electrical charged particles. Different ionization techniques are selected depending on the properties of the compound. Important ionization techniques include electron ionization, electroscopy ionization, and atmospheric pressure chemical ionization. Electron ionization. Electron ionization or EI is commonly used in GCMS. In electron ionization, high energy electrons interact with gas phase molecules. The molecules lose an electron and forms a molecular ion. Because the process provides relatively high energy, the molecular ion can undergo fragmentation. These fragment ions create a characteristic pattern that can be used for compound identification. Electrospray ionization. Electrospray ionization or ESI is widely used in LCMS. In ESI, the liquid sample passes through a capillary to which a high voltage is applied. This produces a spray of highly charged droplets. As the solvent evaporates, the droplets become smaller and eventually produce gas phases. For example, a molecule can accept a proton and form a protonated ions, represent as M plus H in brackets, with a positive charge. ESI is considered a soft ionization technique and is particularly useful for polar, thermally unstable, and larger molecules. Positive and negative ionization. Electrospray ionization can operate in positive and negative ion modes. In positive ion modes, molecules commonly accept a proton and form positively charged ions. In negative ion mode, molecules can lose a proton and form negatively charged ions. The appropriate ionization mode depends on the chemical properties of the analyte. Atmospheric pressure chemical ionization. Another important ionization technique is atmospheric pressure chemical ionization or APCI. In APCI, the sample is nebulized and vaporized. A corona discharge generates reagent ions. These reagent ions then interact with the analyte molecules and produce analyte ions. APCI is particularly useful for relatively small and less polar compounds. Ion optics. After ionization, the ions are guided toward the mass analyzer by ion optics. Ion optics use electrical field to focus, accelerate, and direct the ion. Their purpose is to efficiently transfer ion from the ion source into the mass analyzer while minimizing ion losses. Mass analyzer. The mass analyzer is the heart of the mass spectrometer. Its function is to separate ions according to their mass to charge ratio. Different type of mass analyzer use different physical principles. Common example include the Quadrupole, time of flight, ion trap, and orbit trap. Each analyzer had its own advantage in terms of mass range, resolution, sensitivity, speed, and application. Quadrupole. A quadrupole mass analyzer contains 4 parallel rods. Electrical fields are applied to these rods under specific electrical conditions. Ions with a particular mass to charge ratio have a stable trajectory and pass through the quadrupole. Other ions become unstable and are removed from the ion path. Quadrupoles are widely used because they are robust, reliable, and highly suitable for quantitative analysis. Time of flight. Time of flight or TOF separates ions according to the time they take to travel through a flight tube. Ions are accelerated and given approximately the same kinetic energy. Lighter ions generally travel faster while heavier ions travel more slowly. The measured flight time is then used to determine the mass to charge ratio. Ion trap is an ion trap mass analyzer. Ions are confined using an electrical field. The instrument can selectively inject ions according to their mass to charge ratio. Ion traps can also perform multiple stages of mass spectrometry, making them useful for structural characterization and fragmentation studies. Orbitrap. The Orbitrap is a high resolution mass analyzer. Ions are trapped around a central electrode. Their oscillation frequency is related to their mass to charge ratio. The instrument measures these oscillations and converts the information into accurate mass to charge value. Orbitrap instruments are particularly valuable when high mass resolution and accurate mass measurements are required. Detector. After separation, the ion reach the detector. The detector converts the arrival of ion into an electrical signal. The intensity of these signals is related to the abundance of the ions. The data system then processes these signals and generate the final mass spectrum. Understanding a mass spectrum. The final result is called a mass spectrum. The horizontal axis represents the mass to charge ratio or m by z. The vertical axis represents the relatively abundance or intensity of the ions. The most intense peak is called the base peak and is assigned an intensity of 100%. Other peaks are displayed relative to the other peaks. The patterns of peaks can provide valuable information about the compound. Molecular ion. When a molecular ion is observed, it can provide information about the molecular mass of the compound. Fragment ions can provide additional information about the molecular structure. Therefore, the combination of molecular ion information and fragmentation patterns can help identify unknown compounds. Tandem mass spectrometry. Now let's move to tandem mass spectrometry, commonly called MSMS. In tandem mass spectrometry, a specific precursor ion is selected. The precursor ion enters a collision cell where it interacts with the collision gas. The precursor ion receives energy and fragments into smaller product ions. These product ions are then analyzed. This additional level of analysis provides excellent selectivity and structural information. Collision induced dissociation. One commonly used fragmentation process is collision induced dissociation or CID. The selected precursor ion collides with an inert gas. Energy is transferred to the precursor ion, causing chemical bonds to break. The resulting products ion provide information about the structure of the original molecule. MS versus MSMS. Single stage mass spectrometry provides information about the ions present in a sample. MSMS goes one step further. It selects a specific precursor ion and analyzes its product ions. Therefore, MSMS generally provides greater selectivity and additional structural information. Mass spectrometry applications. Mass spectrometry have a wide range of applications in pharmaceutical. It is used for impurity identification, degradation product characterization, trace level analysis, and structural elucidation. In bioanalysis. In bioanalysis, it is widely used for the quantitative measurement of drugs and metabolites in biological samples. It is also used in environmental analysis, forensic science, metabolomics, and many areas of research. Advantage. Mass spectrometry offers several major advantages. It provides high sensitivity and excellent selectivity. It can provide molecular mass information and through fragmentation structural information. It is capable of detecting compounds at very low concentration. These advantages make mass spectrometry an extremely powerful analytical technique. Limitation. However, mass spectrometry also has certain limitations. Metric effects can cause ion suppression or ion enhancement. The instrument can be expensive and require regular maintenance. Method development may be complex. Proper sample preparation, chromatographic separation, instrument optimization, and data interpretation are important for obtaining reliable results. Complete workflow. So let's summarize the complete process. First, the sample enters the mass spectrometer. The analyte molecules are converted into ions. The ion optics guide and focus these ions. The mass analyzer separates the ion according to their mass to charge ratio. The detector measures the ion. The data system processes the signal and generates the mass spectrum. Finally, the spectrum is interpreted for compound identification, structural characterization, or quantitative analysis. Mass spectrometry transforms chemical information into measurable mass to charge information. From ionization to detection, every stage contributes to the final mass spectrum. Its combination of sensitivity, selectivity, and structural information makes mass spectrometry one of the most powerful analytical technology used in modern science and pharmaceutical analysis. If you found this video helpful, don't forget to like, share, and subscribe.